Hi, everybody. Thank you very much for the invitation to participate in this journal club. As Sylvia was saying, my name is María del Río-González, and I am an assistant research professor at the Department of Psychological and Brain Sciences at the George Washington University, and I am a member of the Social and Behavioral Sciences core of the DC CFAR. And today I'll be presenting or discussing this article, Disparities in the PrEP Continuum for Trans Women Compared to Men Who Have Sex with Men in San Francisco, California, results from population-based cross-sectional behavioral surveillance studies. So just as an introduction, today I'm going to be talking broadly about the inclusion of transgender women in HIV prevention research. And I will start by talking about why I selected this article. Then I'll present an overview of the article, and I will end with a few implications for PrEP research and HIV prevention research more broadly. So first, let's start by clarifying why is it that it's important to study transgender women when in HIV-related research. HIV surveillance systems generally use assigned sex at birth to determine transmission categories, and as a result, transgender women are included under the male-to-male sexual contact rather than as a separate group. Thus, we don't have accurate estimates of HIV prevalence for this specific group because they're just collapsed in terms of the surveillance data with men who have sex with men. However, there is some research showing that transgender women bear a disproportionate burden of HIV. For instance, the most recent National HIV Behavioral Surveillance Survey in 2019 showed that 42% of transgender women with a valid HIV test result from NHBS tested positive for HIV. Although there are some more conservative estimates, for example, a systematic review of 88 studies conducted in the US between 2006 and 2017 showed a 14% lab-confirmed estimated pooled HIV prevalence among transgender women and a 21% self-reported HIV prevalence for this group.
Excuse me, Anna Maria, do
Yes,
you want the slides moved forward or are you handling them?
Oh, they're not moving?
No.
Oh, interesting. Why are they not moving?
Vivian, do you have to move them or what?
No, I should be able to move them.
There we go. There we go.
Okay. Sorry about that.
That's all right. Thank you.
So despite this high HIV prevalence, transgender people in general and transgender women in particular are rarely the specific focus of HIV-related research. And because of this high prevalence of HIV, particularly among transgender women, there have been several calls to identify and respond to the specific factors that contribute to their increased HIV risk, HIV vulnerability, such as this call from the World Health Organization in 2014. And as a result of these calls, inclusion of transgender people in HIV-related research is increasingly receiving global attention, and more studies are including and identifying transgender participants, because it is sure that transgender participants have participated in HIV-related research from very early on in the epidemic. But since gender identity was not asked in the early studies, then we were not able to identify them and as participants in HIV research. So now we are improving over those early years, and we are intentionally including and identifying transgender participants more. However, to be really able to understand and intervened on the factors that heighten HIV risk among transgender people, we need research that goes beyond simply including transgender participants and rather focuses on the specific experiences, needs, and characteristics of transgender populations. Unfortunately, many studies make transgender participants, even though they include transgender participants, they make them invisible when they collapse them with non-transgender groups, which is very frequently the case for research that includes transgender women, but then subsume them under the men who have sex with men category. So we are currently working on a systematic review about how transgender people are being included as participants in HIV prevention research. We have identified 604 studies, of which over half had been published in the last 5 years. We classified these studies into 3 types, depending on how they included transgender people as participants. So our first group is what we call trans-subsumed, and these are studies in which transgender and cisgender participants are combined, most frequently by considering transgender women within the men who have sex with men category. In our review, these studies represent 40% of all included articles. And as we said before, this type of research is an improvement over research that excludes transgender populations or ignores them and fails to identify them properly. But trans-subsumed research has this underlying assumption that results are equally applicable to transgender and cisgender participants, which is frequently not the case. So for example, the case of PrEP is a good example of the potential limitations of this assumption. So a subgroup analysis of transgender women who participated in the IPREX trial indicated no effectiveness on an intention to treat basis for this group, compared to the overall effectiveness of 44% in the IPREX trial. So there is something, the researchers that conducted this study said, okay, there's something going on among transgender women that is making them less, or they are not benefiting to the extent that men who have sex with men are benefiting from PrEP. And the lack of effectiveness among transgender women was associated with reduced tenofovir concentration in blood among transgender women compared to MSM. A secondary analysis of IPREX data showed that there were significant differences in baseline characteristics between transgender women and cisgender men who have sex with men. So for example, depression, transactional sex in the last 6 months, and those baseline characteristics were associated with the differences in PrEP efficacy among those 2 groups. So these findings showed that we need to go beyond simply including transgender participants in research to just really seeing what is about this subgroup or this group that heightens their HIV risk. So we have trans-exclusive research in which only transgender participants are included. And this type of studies represented another 40% of the studies we included in our review. Trans-exclusive studies are particularly useful for identifying transgender-specific HIV prevention needs and for developing or adapting interventions that respond more effectively to those needs. And the last type of studies that we defined in our review is trans-comparative. So in trans-comparative studies, transgender and cisgender groups are compared with each other. And the article that I selected for this journal club was one of the trans-comparative studies that we included in our review. And I selected it because I think it is a great example of how trans-comparative research can be useful for detecting health inequities and for understanding the factors that are associated with those inequities. So with that very large introduction, I'm going to start talking about the article that I selected. So this is an article in which they used two population-based behavioral surveillance studies conducted in San Francisco between 2016 and 2017. The transnational study surveyed 369 transgender women, and the national HIV behavioral surveillance study, the MSM cycle, had 399 participants for that year in San Francisco. The analysis in the study focused specifically in comparing PrEP continuum indicators between transgender women and cisgender MSM, and they found that that transgender women were significantly less likely than cisgender MSM to be at all stages in the PrEP continuum. So they were less aware about PrEP, less likely to have talked with a provider about PrEP, to have used PrEP, and to be PrEP adherents. They also found that although 53% of cisgender MSM could be considered PrEP candidates based on CDC criteria, only 16% of transgender women met this criteria. Specifically, fewer transgender women reported any male sexual partner in the last 6 months, or reported one or more PrEP candidacy criteria. And I think this is particularly relevant because it shows two things. First, there is a need for PrEP candidacy criteria that's specific for transgender women, as currently they are assessed based on the criteria that the CDC has put out for cisgender MSM. And to this point, the authors of the article argue in favor of offering PrEP to any transgender woman who is interested on it and has no medical contraindication because of the high prevalence and incidence in this population. The second thing that this shows is that PrEP might be particularly appealing and useful for those transgender women whose sexual risk is more sporadic and thus might be less interested in taking PrEP for extended periods of time. In addition to the differences that they identified in the PrEP continuum, the authors also identified significant differences between transgender women and cisgender men who have sex with men in terms of several social determinants of health. Specifically, transgender women were less educated, had lower incomes, and had higher levels of unemployment, incarceration, and homelessness. In addition, although transgender women were more likely to have health insurance, mostly publicly funded health insurance, they were less likely to have seen a healthcare provider in the last 12 months, which might be associated with previous experiences of stigma and transphobia in healthcare settings. So even though they have the means to see healthcare providers, they are less likely to actually go to the healthcare providers. And we have to understand what are the factors that are driving that tendency to not go to their healthcare providers. So I think the key message for the purpose of our talk today is very clearly expressed in this selection from the article. Although transgender women often are conflated with MSM in HIV research and services, there are distinct population differences that are important for implementing effective HIV prevention. Our study points to the need for differentiated PrEP implementation strategies to meet the barriers trans women face that are different from MSM. And I think one of the nice things about this article being on PrEP is that in the case of PrEP, the differences between transgender women and MSM are at all the levels. So there are differences there might be differences at the anatomical level, at the hormonal level, at the psychological and behavioral levels, and definitely, as they found out in this research, at the social and structural levels. And so it is important to understand those differences and understand the specific characteristics of transgender population if we want to develop interventions that are effective for HIV prevention among these populations. So I want to conclude the presentation with some take-home points about the inclusion of transgender participants in general and specifically transgender women in HIV-related research. So my first recommendation would be to include transgender people as participants in research. It is very frequently argued that transgender populations are too small for inclusion in research, particularly in quantitative research, where issues of statistical power drive many decisions regarding sample size and composition of our samples. However, in the US, it is estimated that around 0.6% of adults identified as transgender. And so although the population is small relatively to the general population, it is certainly not inexistent. Furthermore, of course, the size of a population shouldn't be an indicator of its importance, and it's a very poor criterion for research inclusion. My second recommendation is to identify transgender people properly. So on November 2017, Dr. Kang led a journal club on an article about using multi-step questions in measuring gender identity. The authors of the article are Greta Bauer and her colleagues. And I think this is a great resource for people wondering how to properly identify transgender participants that are part of the research studies. Also, beyond just including and identifying transgender participants, I think it is important that we are intentional in thinking how and why we are including transgender people in HIV research. But if we think about how we will analyze their data, and based on our specific research questions, we might want to select between a trans-exclusive study in which we only include transgender participants, or a trans-comparative approach, such as the one that the authors of this article took. Researchers who already have trans-inclusive data sets or data sets with information for transgender participants can also contribute to increasing our knowledge about HIV care, treatment, and prevention among transgender populations by conducting secondary analysis that either focus exclusively on their transgender participants or compare them with other groups. And if the sample size is too small for meaningful analysis, then the researchers might consider adding their data to repositories, to data repositories specifically focused on transgender populations, or reporting their trans-specific findings as supplemental materials so that people conducting systematic reviews or meta-analysis can look at that data and include those results as part of our increased knowledge about the population. And the last recommendation that I would have is to make research trans-affirmative and trans-friendly. And this starts by listening to the community and by involving transgender people as part of research teams and or as part of community advisory boards, and really listening what they have to say at all stages of our research. So from the very, very beginning, when we start thinking about, okay, this might be a study that could either focus specifically on transgender people or that could include transgender people, then transgender people should be invited to be part of those research. And it's also important to train cisgender researchers and research staff on transgender health issues and transgender cultural competency. And there are resources such as the Cross-Network Transgender Working Group from the NIH, who developed a series, an online series that is an excellent training source for researchers interested in including transgender populations on their studies. And with that, many thanks. I think I completed my time.
Thank you, Anna Maria. That was very interesting. All right, let's go and let's see, well, perhaps Ruth, from the clinical and population science perspective, what would you like to add or say? Ruth, are you on mute?
I was on mute. Yeah.
Okay. You are on now. All right.
I'm on now.
Do you need the screen or not?
No, I'm good. I don't need the screen. Good. So I had written some notes, so I'm probably just going to read off of my notes. And then first, let me just introduce myself for those who don't know me, and then just briefly about the research that I'm engaged in. So my name is Ruth Kanthula, and I'm a pediatric infectious disease doctor here at Georgetown. And so my work comprises of seeing children with infections in general, and then I also take care of HIV infected children. And then the research project that I'm working on right now is looking at cardiovascular risk in children or young adults who are perinatally infected with HIV. So that's just briefly about me. After reading this article, I think the article did a good job of highlighting the potential targets of intervention, as was mentioned, with regards to transgender women, with regards to their awareness or access and uptake to PrEP. And then I also think it did a good job, as mentioned, in pointing out the differences in the healthcare needs of trans women versus trans men, which may not be intuitively obvious. And as a clinician, the things that stuck out to me with regards being relevant to my clinical work specifically is I think that similar considerations should be made for LGBTQ adolescents and young adults. In this paper, the median age for the MSM group was 36 and 37 for the trans women group, which is slightly older than the children or the population that I take care of. So making sure to include adolescents and young adults in research moving forward, I think is also important. One of the things that stood out to me was the importance of engagement in care. And so and this is with regards to PrEP. And so I think engagement is important because PrEP is not just a one-time thing. And so agree that we should determine or have research that that is looking at figuring out what factors are driving trans women to not engage in healthcare. And so some of them were listed in the paper. And that was the unemployment, those being the sociodemographic barriers, which included unemployment, education, housing, incarceration, and then financial constraints. And the paper did a nice job of pointing out how these factors individually or together could hinder a transgender woman's ability to establish care. And then even if she has insurance, there are issues or there are still continued barriers to care, including past discrimination. So figuring out these factors, I think, is important. And then once a woman is, a trans woman is in care or engaged, depending on how you define what engaged looks like, I think there's also opportunity there for research to actually look from a trans woman's perspective, what her healthcare needs actually look like and what works for her or doesn't work. And the reason that I think this is important is because I imagine that a trans woman's healthcare is a little bit more complicated than what you would expect for an MSM or compared to a heterosexual woman or a lesbian woman, in that it's multidisciplinary and it could look different in different settings, but meaning that there's a PCP, an endocrinologist, a mental health specialist, a gynecologist, or a surgeon. And one interesting thing about all of this is that it allows for some degree of continuity in terms of their care, meaning that they're potentially seeing providers every 3 to 4 months, which gives providers, or there are multiple points of contact within her network where she can either be offered or made aware of PrEP. And then there's an opportunity for follow-up. Multidisciplinary approaches from a provider standpoint also look very different. And so it would be good to just understand what that looks like for the patient and the provider. And the reason I say this is just speaking from a pediatric standpoint. This is just an example. Sometimes I do take care of complex care kids, meaning that they see multiple doctors, but sometimes all the doctors are not talking to each other, for example, when it comes to vaccines. And that's just the simplest example I can give, meaning that maybe the PCP is not giving the vaccines because they're afraid that the vaccines will interfere with their existing condition. And then the doctor who manages the primary existing condition is not giving the vaccines because they're assuming that the PCP is giving the vaccines. So, I mean, I think it just speaks on the importance of understanding how important it is to actually pay attention to what the care that individuals need, and recognizing that we all don't fit into one bucket in terms of our care. And so that was my take on the paper from a clinical standpoint.
Thank you, Ruth. Thank you very much. Amanda, would you like to introduce yourself and then give us your impressions? Amanda, are you on mute?
Sorry, if you don't mind to give me the host so I can share my screen, I would like to present a few slides. So let me see if I can do, oh, perfect. Perfect, thank you very much. So my name is Amanda Macedo. I'm a research scientist in the Alberto Bosque Laboratory, in my team Department, School of Medicine and Health Sciences at the George Washington University. And also I'm a manager of the multiparametric flow cytometry basic science core at the CFAR. Thank you, Dr. Silver, for the invitation, and also Ana Maria for presenting this interesting paper. So from a person from basic science, I'm gonna, I think I have to click to pass my slides. So definitely transgender women cannot be considered as men for different reasons. So there's some characteristics that can impact the biology of transgender women. First, because it's very common that they use feminizing hormones, is the estradiol, and also some transgender women, they pass by the surgery and they have new vaginal tissue. So as a starting article, I found that transgender women on oral HIV PrEP and that are treated with estradiol in a consistent use, they have found that the levels of PrEP, which is Truvada, they both, the two compounds found in this treatment were around 20% lower in the plasma of these women compared to cisgender men. And the PrEP did not affect the hormonal concentration. So for basic science field, we do a lot of in vitro experiments and is in vivo. And it's important to keep in mind that different types of cells can uptake the PrEP in different ways. And I found here an example that was actually studied in women, in cisgender women. So what they did was they took tissues from the female reproductive tissues and also from blood, and they treated the cells with tenofovir, which is found in the PrEP, and they analyzed the concentration of tenofovir inside the cells, the active form of this drug, and they found that there's a variability of this active compound across the cells. So epithelial cells and fibroblasts, they had a higher concentrations compared to CD4 T cells, the lymphocytes, which are the main targets for HIV, both in the tissues and also in the blood. So this shows that there's a difference in the uptake of the PrEP. And in the same study, they found that treating at the same time progesterone and estradiol decreased the intracellular concentrations of the compound. So here in this graph, they show that only estradiol did not alter the uptake of tenofovir by the cells, and combining estradiol with progesterone had led to less uptake of tenofovir by the cells. So unfortunately, these studies were done with female reproductive tract, and there's very few data, limited data, showing what would be the effects of using testosterone and also treating male tissues or cells with female hormones. And as a basic science researcher, I would like to bring the awareness of some factors that need to be taken into consideration. For example, the assigned sex of birth, and also the gender, the current gender of the donors from the samples that we use. Also, since we do a lot of cell culture, we need to keep in mind and use the right specific reagents and media in our culture. We are aware of this in our lab. We know that some reagents, for example, serum, we have to use specific charcoal strip serum that are depleted of hormones to do these studies to not have confounding factors. Also, we need to wash the cells in order to have, to do the culturing. And a lot of times the hormones are washed away. So we need to treat with exogenous hormones, and also to, if you want to understand the effect of changing that hormone and seeing the effect of addition of that hormone on the female or male cells, what would be the consequences? And we have to keep in mind of the cell type that we're using in regards of the variability of expression of hormones, and also the source of tissue. As I mentioned in the paper that I showed, the figure I showed, depending on the cell, they can express and uptake differently at PrEP. So that's very important to keep in mind. And we're talking about transgender women, but also we need to keep in mind that there's a lot of articles on female and using female tract, but also we need to include transgender male in their studies. And transgender women are not men, as I mentioned before, and they are not women either, because in regards of their biology, we need to control this to include in our studies. And yes, so that's basically what I would like to share today.
Thank you, all of you. That was really very interesting. I mean, it's interesting how you all came at the article from your different points of view, which is exactly what we want to try and bring out during these journal clubs. The fact is that the things that were brought up that, you know, from my point of view, I'm listening and, you know, the importance of engagement in research. I mean, that's something across the board because there's so many different things that people's lives are involved with that researchers, you know, they're looking for people. They have absolutely no idea in some cases what that person's day even is like, rather than what their history, what their physiological balances or things like that. And a lot of what was said today impacts just on research in general and talking and working with participants. But let me open it to to everyone on the call. This is the time now to ask questions, to make comments and things like that. One thing, Anna Maria, is very interesting. There are data sets out there that people can use. With research on transgender women, I imagine it's very small. You said NIH has one. Is that something that, you know, we could make sure everyone knows about? That if, you know, you're doing a research project and you don't have a significant number, that you can maybe get some data from other studies to boast your power?
So there are some data. For example, the US trans study, they have data and they have it in the ICPR's data repository. And there are a few other studies. Most of them are trans-specific. So would only include transgender people. But just collaborating with other researchers, and I think that that's also one of the reasons why adding this findings, even as supplementary materials, might help connect with other researchers that are conducting similar research in order to pool data and get a better sense in terms of numbers, but also a better sense in terms of differences by geography or over time periods. So just being intentional in the way we talk about findings that are specific to transgender participants in our research, I think that motivates other researchers to think, oh, this is something that could be done, and that's important to do. And regarding the NIH, what they have is a training, it's like an online training module for HIV, for research that includes transgender participants. And they have sort of like from the very, very, very basic issues in terms of language, in terms of cultural competency, to more complex issues related to what Dr. Kanthula was talking about and Dr. Macedo as well, in terms of biological, medical, social, structural conditions that are specific for transgender populations.
And so, you know, Amanda, when you brought up, you know, the fact of hormone levels having impact on that, you know, I wonder how many basic researchers actually know more of the medical history of the participants that they may be using specimens from, you know, because the right hand might not know what the left hand is doing, but also there may be some sensitivity, I wouldn't say training, but some sensitivity that should be given out to basic scientists about more knowledge about the specimens and the history of the participants, or making their targeted collections more, I guess, targeted, you know, because that was very interesting.
Yes, exactly. And there's more and more awareness of how much hormones can impact not only like the PrEP, but also HIV infection and transcription. And actually, the NIH is requiring more and more to be aware of even the sex differences, because sometimes even, specimens are not distinguished between male and female, but it's actually much more complex than that. And yes, this is I know how sometimes challenging can be to gather samples from patients, since we have limited numbers, but it's very important to keep this in mind, that the hormones and even the gender of the person, the sex, can impact in the research.
This is a very interesting topic, and of course, one of interest to NIH as well as research into that. And Alan's raising his hand. Yes, Alan, but you're on mute. There you go.
Thanks, Sylvia. Sorry, I'm out and about today. We just learned this week that there'll be a new inter-CFAR working group on transgender science that will be led by the University of North Carolina CFAR, and that all of our investigators and community members who are interested in the transgender health will be able to join. So Patty will be disseminating information about that next week, but that's very exciting and a sign of the commitment that NIH has, I think, to the population that you were referring to. So I just thought I'd add that in.
Thank you. That's very good news. Very good news. So everyone who's on this and has any interest in that, make sure that you watch your email for Patty's announcement. Does anyone else have any comments or any questions for our presenters? No. Well, the thing is, when you were looking at the lack of adherence, that specifically, when Amanda was talking, I'm wondering whether or not some of that isn't lack of adherence. If they're seeing low levels, when they're seeing whether or not, when they think people are, taking the medication, if it isn't a fact that, it isn't that they're not taking it, it's that their levels are low because it isn't doing as well in that person as possible. So data showing lack, of adherence may be erroneous. That's something else to be thinking about. I wonder if there's any thoughts on that, or any kind of research that we could be doing that would be able to, pinpoint or look at things like that when people are thinking about, PrEP and how much it is actually being used. Ruth, have you thought of that in looking at, do you measure adherence from talking, social, or actually trying to measure the therapeutic level in someone?
So I usually measure adherence from talking. And so, and I'll be honest, that's adherence with regards to HIV medications, because I don't have too many teenagers in my clinic. It's a small clinic on PrEP. But I think that was one of the thoughts that I had when I was reading this paper, would be to, again, what does measuring adherence look like for this population, given their, I don't want to use the word aversion, but that the fact that going to or being in contact with a medical provider can be triggering in some fashion. So, maybe you miss a pill because you didn't go to the doctor to pick up your pill because you don't like going to that doctor's office because every time you go there, you feel ashamed or something like that. So it would be interesting if there was a study where you could actually consistently monitor someone's adherence, I guess, biochemically, and then also compare it to their reported adherence and see what that looks like. Because I wonder if it's just a combination of the two. I'm missing my pills because I don't want to go and deal with the healthcare system.
It could be, but some good research showing, because it's also the perception of the population that they won't be adherent to taking PrEP, whereas maybe they're more adherent than people feel they are. It's just that biologically it isn't showing up that way. That would be something to do. That would be a research thing that I think would be very interesting to the HIV research community, is to again show that this perception may be or may not be wrong, because actually they are doing it, they are taking it, but they're just not getting the therapeutic levels that they should be taking.
Yeah, there are actually very small studies in which they monitor daily uptake of PrEP, and they also monitor the use of hormones. And I think this is one of the studies that Amanda was talking about at the very beginning. They do show that beyond the issues associated with adherence, that there are some issues associated with adherence, as Ruth was talking about, social structural issues and experiences with the healthcare system, but also issues of homelessness, income. There are other elements that might be more urgent for transgender persons. So adherence might not be very high in those cases, but even where adherence is monitored, there is a drop in blood concentration that is associated with taking the hormones. So, and that is one of the reasons why non-daily PrEP is not indicated in the case of transgender people, because the levels, the research shows that for men who have sex with men, for cisgender men who have sex with men, 4 days a week can be protective. But for transgender women, those same 4 days might not be protective. So there are some implications in terms of how we clinically manage PrEP for this population.
Very interesting. Very interesting.
Yeah, absolutely. The mechanism of action. So I've been reading a little bit. I was very interested in trying to understand the mechanism why the transgender women, they have lower pharmacodynamics, and they believe that there's a higher renal clearance, but they don't know how. So those the PrEP is just eliminated out of the body faster compared to cisgender men. So it's not known. So yeah, it would be interesting. So but those therapeutic approaches that like, for example, cabotegravir, maybe they should include, but decrease the window of administration compared what is current, given to people. So yeah, there's definitely more room for studies.
Very interesting. Very interesting. Any comments from anyone? We're getting close to the hour.
The only additional comment I had, just piggybacking off of what Amanda said, is perhaps maybe there is need for a different combination of drugs for PrEP for transgender women, instead of focusing on what we know may not potentially work, looking at other drug combinations that might work for them.
Another good area of possible research to see. All right. Well, I want to thank our presenters and Anna Maria for thanking you for choosing that article, and Ruth and Amanda for responding. I hope it has sparked some thought or interest by people to look into this. Amanda's brought in some interesting things, and I'm sure there's an area of research out there that just needs to be filled. So anything at all before we end? No? Well, thank you. Thank you very much. And yes, I agree, Maria Cecilia, great discussion. Take care, everyone.